Right, class, let's go ahead and get started this morning. Good morning, everybody. Hopefully you guys found that exam not to be too difficult. I feel like I made it relatively fair. If you have any questions about the exam, feel free to see me during office hours or after class. But the majority of you guys did really well. 87% was the average, so I'm pretty content with that. Hopefully the final will be something similar, if not better. But basically the next half of the class is going to be focused on the immunology aspect of things and macromolecules of life. So this week, hopefully, is going to be not too intense. I'm going to go ahead and layer on some of these complex ideas like I did during the first week when we're talking about the central dogma and DNA replication. So if you guys have any questions, feel free to raise your hand or put it in the chat. So today we're going to be focusing on immunology, more specifically, one of the arms of the immune system, the innate immune system. So what I'm going to do first is basically give you guys some key definitions that you guys should know, because I'm going to be using them quite a bit throughout the lecture. And if you guys don't have a full grasp of what those words mean, then you might struggle a little bit. So you guys all have access to the lecture slides. So if if there's a word I'm saying over and over again and you're not sure what it is, refer to this next slide. I'm going to give you guys a brief overview of the immune system, but then I'm really going to focus on the innate immune system, and tomorrow will be more of the adaptive, and we'll go and connect those two. I'll give you guys a little bit of a preface on the connection between the two today, but to not overwhelm you guys with the material, we're going to focus on the adaptive, the B and T cells tomorrow. I'm also going to be going over the differences between the primary and secondary lymphoid organs. And so this is where immune cells are going to originate and live. So understand, what's the main difference between those two. And then I'm going to highlight some key innate immune cells that you guys should know. I will be asking you guys questions on the exam about that. You'll get a little bit of a taste of that today when I give you guys some the lecture quizzes to see what I'm going to be asking. And then what I really I'm going to end up going over is how the innate immune system recognizes foreign invaders. And so the main learning objectives are basically what the outline is: know the terms, know the difference between innate and adaptive immune cells, be able to distinguish who's who, know the difference between the primary and secondary lymphoid organs, know the receptors that are implicated in recognizing foreign invaders for the innate immune cells, and then know the difference between chemokines and cytokines. And if you're unfamiliar with a lot of these terms, hopefully by the end of the lecture, you will understand. So here are going to be a list of key terms that are going to be thrown around a lot this lecture. So the immune system. The immune system is going to be a set of tissue, cells, and molecules that mediate resistance to infection, whereas immunology is going to be the study of the immune system. Immunity is going to be the resistance of a host to pathogens and their toxic effects. So basically, people or organisms being able to defend against foreign invaders. The immune response is going to be this collective and coordinated response between the innate and adaptive immune cells, coordinating with cells that are infected to go ahead and mediate a response to get rid of these foreign invaders or these pathogens. The pathogens are going to be microorganisms that cause disease, and antigens are going to be foreign non-self molecules or substances that activate the immune system. So these are going to be things that aren't naturally found in our bodies. And then vasodilation is something I'm going to talk about at the end, and I'll try to reemphasize it, but just to give you guys a preface to it, vasodilation is when your blood vessels are expanding, and it's something that your immune cells regulate when infection is occurring. So why do we even have an immune system? Well, the immune system has a lot of vital roles in our body. When it comes to tissue homeostasis, or basically the equilibrium of our body, or keeping this balance in our body, the immune system is really good at removing dead or damaged cells. So I talked about before when Cells are mutated, they undergo some of these checkpoints, and they get programmed for cell death, so apoptosis. Well, when these cells undergo apoptosis, they end up fragmenting or blebbing, and they're not just free floating in your blood and stuff. Your immune system actually comes in and basically eats all of that up. Your immune system also plays a critical role in killing cancer cells. So it's thought that, every day we're developing cancer cells, but your immune system is good at checking to see if they are cancer cells, and that's mediated through some receptors that cancer cells will end up expressing on their cell surface, and the immune system will be able to basically detect them. Another thing that you might not be aware of is that your immune system plays a role in wound healing. So whenever you get a cut or a bruise or something like that, your immune system's coming in and trying to heal that area. And that's through a variety of molecules and immune cells. Basically, one thing is, whenever there's damage, there's always going to be some kind of cell debris present. And the immune cells are going to be there to kind of clean that up. If it's a cut from the exterior, you're sometimes going to introduce bacteria into the wound. And so there, your immune system is going to be there to kind of fight off the bacteria and keep it clean. And then on top of that, your immune cells actually play a role in promoting clotting. So the wound actually heals and produces other molecules that actually promote the abscess or wound to actually regenerate. And then the main thing that we're going to be focused on in this class is tissue defense. So as you guys should all be aware of, your immune system is really important for preventing pathogens from even getting into the body. But in the event that it does get into the body, your immune system plays a role in killing off those pathogens and removing any toxins that may have been released from the organism. Your immune system is really good at preventing that organism from spreading, but it's not perfect. Sometimes, the organism might get away and travel somewhere else and then, create more inflammation in other areas. But overall, your immune system is good at preventing it from spreading. And then surprisingly, your immune system is good at, we all have, our own healthy bacteria, but somehow the immune system doesn't recognize it as a pathogen and recognizes it as their own, which is kind of cool. So what does the immune system protect us from? It protects us from a variety of pathogens that cause disease. And here I have a couple of listed ones that are present. And so, extracellular bacteria, parasites, and fungi are going to be common ones. And depending on the size of the pathogen, it's going to be dependent on what kind of immune response is mounted. I'll get more into depth about this tomorrow, but just as a preface, usually cells that are pathogens that are too big to invade a cell, usually the main immune cell, adaptive immune cell that's going to play a role in fighting against those bigger molecules are B cells, which secrete antibody. And that's so other immune cells can come and try to eat it. As opposed to intracellular organisms like intracellular bacteria, parasites, or viruses, as we've covered, they all need a host cell to replicate in. And so with this particular immune response, it's mainly going to be cytotoxic T cell mediated. So basically, if the cell is infected, to prevent the pathogen from having a nice home to replicate in, your CD8 T cells are going to come in and basically kill these guys so they can't replicate. And then same thing with the extracellular worms that go along with the extracellular parasites and fungi. Usually it's going to be B cell mediated because they're just too big to get into a cell. So your body's just trying to excrete it out. So the first line of defense before the immune system even recognizes a pathogen are going to be our barriers. And so we have three different kinds of barriers that we have in our bodies to keep pathogens at bay. And so the first one's going to be the physical barrier. And so these are going to be things that physically are able to keep pathogens out. The most common one is going to be the skin. The skin is going to be there to protect you from, anything getting into your body. But if you do have any cuts or something, then that's going to be a break in the defense, and that's going to allow for pathogens to enter your body. And then you have some kind of cilia in your like esophagus and the trachea area that basically, well, if you end up inhaling some kind of particulates, these are the reasons why you end up like coughing or sneezing. It's because things get trapped in there and they're trying to protect things from going into the lungs. And basically when things get stuck in there, they'll end up kind of moving a bit. And then once there's enough irritation, then you'll end up sneezing or coughing. And then urinary tract infection. Your urinary tract is going to be used to flush out, any pathogens that may have gotten into the bladder area. You also have chemical barriers. And so the chemical barriers are going to be there to really make the environment unhappy for any pathogen to live. So in your tear glands, within your actual tears, you have these enzymes called lysozymes. And so those are pretty acidic, and most pathogens don't like acidic environments. And then the other area are going to be your GI tract, specifically in your stomach. Your stomach, I think, goes to like an acidity of like 1 or 2, which is pretty acidic. And most pathogens don't like to live in acidic environments. So if something gets into your stomach, most of the time the organism will end up being killed because it's just a very unhappy environment to live. But pathogens being how they are, they learn to evolve mechanisms to protect themselves from some of this high or low pH and ends up making it into the intestines sometimes. That's common with a lot of parasite and bacterial infections. And then the last line of barrier defense are microorganisms that actually live within your gut and also on your skin. These are there to outcompete foreign bacteria. And so if you don't have, a good set of bacteria present, then it's not going to be able to keep the bad bacteria at bay, and then you end up getting sick. So keep your gut bacteria healthy by eating good stuff that's going to feed it. So of course I'm not going to have you remember all of these different species of bacteria, but what I want you guys to appreciate with this slide is we coexist with a large number of good bacteria that are in all of these different areas in our body. The majority is exterior, on our skin, on different areas. But then you have some that, once you enter your mouth and follow the GI tract, all of that area also has healthy bacteria that are meant to keep your good bacteria protecting you from the bad bacteria or any other foreign pathogens. And then to highlight, the densities of different bacteria where it's present. Usually in the large intestine is where you're going to have the majority of your different species of bacteria relative to other regions. Let's see. Where do the bacteria come from? It's an interesting question. So, I think it really varies. So we don't live in a sterile environment. So, there's bacteria all around us. We don't see most of it. But a lot of the time it comes from just us touching things. And most of the bacteria that's present in the environment isn't harmful to us. And so there are some species of bacteria that are able to live on us. And I'm sure it has to do with certain like components that we secrete in our skin that keep the bacteria, happy. But there's also, we get some of it from our mom when we're born. So if people don't, if they're born through a C-section, then they don't. But I know that there's studies that have shown that it's good for babies to actually be born like naturally because of some of that bacteria that's present that comes on the skin. It's actually good for us when it comes to developing some of that commensal bacteria. Can there be an instance when our good bacteria can be bad for us? I'm sure there is. I think it has to do when there's overgrowth. And so Like there's always, so we not only have good bacteria, we also have like good fungus. And so I know like when with like females, when they end up, you know, end up taking some antibiotics, sometimes this messes with the pH and the gut microbiota environment in the vaginal area, and that ends up skewing things. And when certain species are not in line or they're not, you know, in balance with the other ones, then, you know, overgrowth can happen and that can lead to problems. So it's important to know that the immune system has two branches. It's the innate and the adaptive immune system. And so I'm going to give you a preface to both of these, but the lecture is going to be mainly focused on the innate, and tomorrow will be focused on the adaptive. So with the innate immunity, basically these are going to be the first responders. These are going to be the immune cells that are able to see pathogens via these pathogen recognition receptors, which I'll go into later. And with these receptors, they're not, they recognize things that aren't ourselves, like, you know, bacteria components or viral or parasitic components, but they're not very specific. They don't recognize like, you know, something very, a specific species. They kind of just recognize, you know, everything. As long as the component isn't found in us, then they'll be able to respond to it. It's also important to note that innate immunity cannot develop memory. So even when it's exposed to any kind of pathogen, it will never develop memory. At least, you know, no scientists have demonstrated that at this point. Now, when you compare this to the adaptive immune system, it responds much later. You need to basically have some kind of cell, usually innate immune cells, present antigen or this, you know, molecule of the foreign pathogen to the adaptive immune cells, so they can get educated and develop a memory response. With adaptive immune cells, they are very specific. They only recognize specific antigens or these protein complexes for them to be able to actually get activated. And we'll get into much more detail about that tomorrow. Here I have a schematic of the typical time it takes for innate immunity to kind of, you know, actually begin. So, you know, once an infection begins, we already have, you know, free floating molecules in us to kind of prevent pathogens from going crazy. And sometimes those are good enough to prevent any kind of infection from, you know, manifesting. And so antibodies are going to be one. You have free floating antibodies in your body that are trying to basically bind to whatever pathogen it's seen before. There's another thing called complement, which I'm not really going to talk about in this lecture because I think it's just too complicated and it's beyond the scope of for this class. But basically you have this thing called complement in your system that are molecules that are free floating that like to bind a pathogen, specifically bacteria. And once it recognizes the bacteria, it'll actually bind to it and form holes in it. So it's easier for the bacteria to die. Now, if for whatever reason the infectious agent isn't removed, then your innate immune system starts to kind of kick in. And that's when it'll start to recruit other immune cells or other innate immune cells to basically come and help and clear the infection. And that's going to be done through a variety of ways, which when we highlight specific immune cells, I'll go over that. And so usually most of the time, this is good enough for the infectious agent to be cleared. Now, if, you know, the infectious agent has been troublesome because it's evaded the immune system a bit or has been replicating in cells and hasn't been detected, then that's when your adaptive immune response takes over. And notice how long this takes. It takes like almost 5 days before, you know, the innate immune system starts to actually talk to the adaptive immune system to start mount a specific response. And so just a preface to this, basically what will end up happening is innate immune cells will start to talk to your adaptive immune cells. And then once it recognizes that specific antigen it was meant to fight against, you're gonna, the B and T cells, which are your adaptive immune cells, are gonna undergo something called clonal expansion. And basically this is just meaning this one cell is going to multiply itself X amount of time. So there's enough of it to go ahead and circulate throughout the body and go and fight. And it's usually when your B and T cells have been educated and have expanded enough that you're able to actually remove this infection agent. And once you've done this, your B and T cells will undergo something called clonal deletion, and basically it ends up contracting where you only have a small population of these cells that are able to recognize a secondary infection. And so these are going to be what your memory responses are. And I know that might be a lot, but like I've done before, I'm going to layer on all of this information as we go on. So you guys have multiple times of exposure to this. So here we have a kinetics of a successful inflammatory response to infection. So here we have on the x-axis the duration of infection, and then on the y we have the level of the microorganism. So here at the starting point we have a microorganism that enters our body, and unfortunately, it does take our body to take some time to recognize that there is a foreign invader. And it's not until it reaches a specific threshold that your immune system starts to recognize that there's something that's present that shouldn't be there. And so during this first stage is when the infection is establishing itself. And then your innate immune system is going to do its best to try to clear it. But if it doesn't, then it's going to recruit the adaptive immune cells to start trying to help and kill off the whatever the microorganism is. Once there is enough of the adaptive immune system educated and there's enough of them that have clonally expanded, then you're basically going to get your peak level of microorganisms, and then the adaptive immune response is basically going to clear this infection. And then once the infection is cleared, then you have memory against that particular pathogen. So if your body were to ever see it again, instead of having to go through all of this again, you already have cells that are educated to go ahead and recognize that pathogen to clear it. And we'll go again, I'll reiterate a lot of this in tomorrow's lecture too, when we're talking about adaptive immunity. So, it's also important to emphasize that both branches of the immune system are needed to clear the infection. And so in yellow, I have that curve that I just showed you in the previous slide. And so this has both branches of the immune system. So it takes some time for the immune system to ramp up, and then once the adaptive immune system is educated, then it'll start to clear it. Now, when you lack an innate immune system, again, these are going to be the first responders, the level of microorganisms ends up increasing exponentially. And that's because you need innate immune cells to communicate with the adaptive immune cells to generate an effective immune response. If you don't have that communication, then your T and B cells are never going to know that a pathogen is present. If you were to put a T and B cell in the presence of like bacteria or something, they're not going to see it. They need some kind of cell to do something called antigen presentation, or take a piece of that pathogen and then present it on its cell surface so the adaptive immune cells can respond to it. And so innate immune cells are very big with presenting antigen to your adaptive immune cells. So if you don't have that, the microorganisms are going to go crazy and you're going to end up dying. And so all this is a graph showing stuff that occurs in mouse lines that people have used to study immunity. In the green line, we have basically only our intact innate immune cells, but we lack an adaptive immune cells. And so, like I was saying, the first responders, so they'll be able to, hold off the infection for some time, but they only can do it for so long. In order to clear this, you need the adaptive part. If you lack the adaptive part, basically you're going to have something called chronic infection, where you're going to have infection for the duration of your life, unfortunately. And, if it gets bad enough, then you can succumb to the infection. So the immune system originates in the bone marrow. So we all have bone marrow, and I think most people probably associate the bone marrow with red blood cells, but this is also where your white blood cells or your leukocytes or your immune cells are located. So all of these white blood cells, leukocytes, immune cells, they're all used interchangeably. They're the same exact thing. And so this is going to be the site where your immune cells develop and mature. And so the main primary lymphoid organ is going to be the bone marrow. And so this is going to be the start site where all immune cells and red blood cells are going to begin. You have this pluripotent stem cell that basically can differentiate into three main cell types. It can turn into a red blood cell. It can turn into a myeloid progenitor cell, which is going to be your cells that generate into your innate cells. Or it can generate into this lymphoid progenitor, which is going to be all of your adaptive immune cells. It is also important to note that it's not only your bone marrow that is considered a primary lymphoid organ, but it's also your thymus that sits right above your heart. And this is where after your T cells are made in your bone marrow, they're actually called thymocytes at this point when they're in the bone marrow. And I'll mention a little of this tomorrow. But in order for them to mature into T cells, they have to go into the thymus. So hence the T from thymus, that's where your T cells originate from. And so kind of going off of that, once your immune cells are mature, they're going to end up leaving into the blood. And like your T cells are going to circulate and then end up in the thymus. If they're some kind of innate immune cells, such as a neutrophil, eosinophil, basophil, or monocyte, they're going to end up circulating throughout the blood to look for foreign invaders. It's basically once a foreign invader has been recognized, some of these innate immune cells, such as dendritic cells or macrophages or monocytes, end up going to the secondary lymphoid organs, these ones highlighted in blue. And this is where antigen presentation is going to take place. This is where your innate immune cells are going to educate your adaptive immune cells to initiate an adaptive immune response. So you have multiple secondary lymphoid organs all throughout your body. So you have the spleen, you have your appendix, you have this region called Peyer's patches that are in your small intestines. You have, your lymph nodes, and in the back of your throat, you got your tonsils. And so in these areas, you basically have a bunch of B and T cells and some innate immune cells that are coming in from the external environment, taking up antigen and presenting it to the adaptive immune cells, B and T cells, to alert them if there is a foreign invader. So here, now we're going to go to layer it on again. So I've already told you that there's two branches of the immune system, innate, which are also going to be the myeloid organ cells, and the adaptive, which are going to be your lymphoid organ cells. You basically have in your innate immune cells neutrophils, eosinophils, basophils, mast cells, macrophages that can differentiate into monocytes that can differentiate into macrophages, and monocytes that can differentiate into dendritic cells. In your adaptive immune system, you have only B and T cells. There are a couple of other immune cells that I did leave out, like natural killer cells and megakaryocytes, but these are the ones that I'm going to end up highlighting in the next 2 days. So be aware of what the function of each one of these is, and have a clear understanding on what their role is when it comes to infection, and in particular viral infection, when I highlight that tomorrow when we're once you guys have a full understanding of what adaptive immunity is. So for the rest of this lecture, I'm going to be mainly focused on the myeloid lineage of cells and highlighting their function, and then I'll sprinkle in some adaptive immunity to kind of link the two, but this is going to be the main focus. So you have three kinds of granulocytes. So granulocytes are just cells that are able to produce granules to basically kill off pathogens. And so the most abundant granulocyte is going to be the neutrophil. And so neutrophils make up roughly 40 to 75% of the total leukocytes or white blood cells that are present in your body. And what they do is they're most associated with fighting against bacteria. And once they recognize the bacteria, what they're able to do is actually capture them by undergoing phagocytosis or engulfing or eating the pathogen. Um, they have effector function, meaning that they can alert other immune cells to basically come and help out with the infection. It's also important that they work in anaerobic or conditions that lack oxygen. And this is really helpful when it comes to sites of severe tissue damage, because some of those lack oxygen. And as you know, all cells in your body require oxygen to survive. And so this is one of the few cells that can actually function under conditions that lack oxygen. They're also very short-lived. And one of the reasons why they're short-lived is they try to prevent um, infections from spreading by killing themselves. And they kill themselves by producing these things called neutrophil extracellular traps, or nets. And so here I have an example of a neutrophil highlighted in yellow, throwing out some of their nets. And what their nets are just composed of are proteins and their own DNA. And this is going to trap bacteria highlighted in blue and prevent it from basically leaving the site of infection so it doesn't go systemic. The other kind of granulocyte are eosinophils. And so this makes up anywhere from 1% to 6% of all your immune cells that are circulating in your body. And they play an important role when it comes to defending against worms. And so here I have an image of a bunch of eosinophils that are fighting against this giant worm. And so notice, compare the size of the immune cells to the worm. The worms are extremely large compared to these cells, and these worms aren't able to actually invade cells because they are so big. So typically they'll end up attaching themselves to the intestine or something to feed off of the host, compared to like some intracellular bacteria or parasites where they require a host cell to live. I won't quiz you guys on this, but just to kind of highlight why they look the way they do. So eosinophils get their name from when scientists were basically taking blood samples and staining immune cells to see what were present. And so there's a stain called hematoxylin and eosin that basically are used to help identify and stain cells. And so the reason why eosinophils are named the way they do is because when they were stained with eosin, they stain red. And this indicates that their granules, or their little, molecules that they're meant to secrete to kill off foreign invaders, are basic inside them. So they're going to, cause the dye to react and stain red. And then the last granulocyte that I want you guys to know are basophils. And so these are also another immune cell that regulate parasite infection. There isn't a whole lot known about these cell types. Most people don't study them because honestly, there's more important cells to study, like B and T cells. But I'm sure where it gets its name from basophil is because of the basic components that the dye ends up staining with. So as I mentioned before, hematoxylin, eosin, the hematoxylin is going to end up staining some of this compartment blue, and that just has to do with the acidic components that are within these granules. The other innate immune cell that you should know are mast cells. So mast cells are the reason why some people have allergies. And it's because these mast cells are the ones responsible for releasing histamines. So, typically people that have allergies will take antihistamines to prevent the secretion of histamines into the environment that causes all of those typical symptoms like runny nose and sneezing and whatnot. And so I'll highlight this again tomorrow when we're talking about antibodies, but a lot of the innate immune cells have these things called FC receptors. And what the FC receptors do is they pick up free-floating antibody that's floating in the blood. And mast cells in particular interact with this antibody called IgE. And so there's 5 antibodies total that our body is able to make, and I'll highlight those tomorrow. But just as a preface, mast cells only recognize and bind to IgE. And so here we have an antibody on the surface after it's bound to the Fc receptor. It's important to note that this antibody has this Y shape. And so the way that these mast cells work are so antibodies are part of the adaptive immune system, and they recognize very specific things because of their binding domains that are on the pointy parts of the Y. And so if it recognizes something foreign, then it's going to end up causing the mast cell to release its granules. And once it releases those granules, these granules are the histamines. And that, again, is going to cause all of the typical symptoms that are associated with allergies. And so one thing that some of the people might be sensitive to is pollen in the air. And so pollen, it is a foreign substance, but most of the time, people that don't have allergies, their antibodies don't really bind to the pollen very well. And so if they don't bind to the pollen very well, then your mast cells aren't going to end up releasing their histamine. But there are some people that are very sensitive to the pollen, and that's because of these antibodies that have a high affinity or like to, bind to the pollen very well. And once that pollen is bound, then it causes the histamines to be released. Another immune cell that you guys are gonna for sure know by the end of this class, because it is very important when it comes to interacting with the adaptive immune system, are macrophages. So as I showed you from the lineage diagram a few slides ago, macrophages originate from monocytes. So monocytes are free-floating, immune cells in your blood. But once a monocyte is recruited to a site of infection or sees that there's a pathogen present, it ends up differentiating or becoming a macrophage. And so these macrophages are going to end up staying in that particular tissue, and then doing whatever it needs to do to get rid of the pathogen. And so I always describe the macrophage as the garbage collector of the immune system, because this is the one that likes to phagocytose objects. So the main things that in a homeostatic balanced environment without any kind of pathogen, they play a role in cleaning up dead cells and debris. But when it comes to fighting against pathogens, it actually ends up engulfing or phagocytosing the pathogen, so it can't actually invade other cells. This macrophage is very, very important when it comes to activating the adaptive immune system. And it does this by 2 ways. It does this by secreting pro-inflammatory molecules called cytokines. And then it also, so those are going to be the molecules that the immune system communicates with one another to go ahead and, say, if it's a pro-inflammatory molecule, hey, get activated, there's infection going on, come and help clear this infection. Or anti-inflammatory molecules that basically, once the pathogen is cleared, then go ahead and just calm down. There's no need for all this, chaos, all these immune cells to be present, go back to where you came from or die. And then the other thing when it comes to the adaptive immune response, or when educating the adaptive immune response, is it does antigen presentation. And I'll emphasize this a lot in tomorrow's lecture, as this is the only way adaptive immune cells, T and B cells, can actually see a pathogen. They only can see a fragment of it by other cells presenting antigen to it. The other really important cell that when it comes to helping to initiate an adaptive immune response is the dendritic cell. So like the macrophage, which they're all innate immune cells, they originate from monocytes, and then before it even leaves the bone marrow, it becomes a dendritic cell. These dendritic cells are called immature when they haven't seen a pathogen yet, but they're usually, some people will describe them as police or the patrol, because they're just circulating throughout your body looking specifically for pathogens. And so once it identifies a pathogen, it ends up becoming a mature dendritic cell. And what it ends up doing is basically taking whatever the pathogen is, so bacteria, virus, fungal infection, it'll take a piece of that and then present it to the T and B cells, and so they can go ahead and initiate an adaptive immune response. So macrophage and dendritic cells are very crucial when it comes to facilitating this process. And so here I have an image, and of course, I'm not going to have you guys know this, at least for this lecture, I might go until tomorrow. But this is basically the process of antigen presentation. You have a dendritic cell that has some peptide or antigen that's on its cell surface that's facilitated through this receptor, which I'll get into tomorrow. And this receptor and pathogen or antigen is going to be how the T cell recognizes or sees the pathogen. And this is mediated through its T cell receptor. And there's a whole bunch of other things that end up going on, which I'll highlight tomorrow just to not confuse you guys. But this process in particular is really important to understand, because this is how the T cell is able to interact with the innate immune cells to see that there is some kind of foreign invader present. If this interaction doesn't occur, the T and B cells will never mount a response against any foreign invader. So how are the innate immune cells able to even get initiated? Well, this is all generated through being able to recognize the foreign pathogen. And this is through these things called pathogen recognition receptors or pattern recognition receptors. People will use these interchangeably. These are going to be PRRs. And what they're actually recognizing are these pathogen associated molecular patterns, or PAMPs. And what these PAMPs are they're conserved molecules on the invading pathogens that are generally not found within our own system. So these are all going to be recognized as foreign. And once that's recognized as foreign, then an immune response can be initiated. And so I have here an example of a type of receptor that is able to recognize foreign pathogens. And these receptors are called toll-like receptors. And so these are expressed on all immune cells, all innate immune cells. And so here you can see an example of what exactly this toll-like receptor is recognizing. There's a couple of different families of them. Of course, I'm not going to have you guys know all those, but just understand that an example of a pattern recognition receptor is a toll-like receptor. And these toll-like receptors are going to end up binding to specific conserved pathogen molecules that are not typically found in humans. And so once it recognizes any of these molecules, then the innate immune cell, whichever one is expressing these, is going to end up initiating an immune response. So what ends up happening when these pattern recognition receptors actually get activated and stimulated? Well, once the immune cell, the innate immune cell, ends up getting stimulated through this receptor, it's going to end up doing two main things. It's going to end up secreting these molecules called cytokines and chemokines. So the cytokines, which I mentioned earlier, are going to be the effector molecules that tell the body, the whole body, not just your immune cells, also your non-immune cells. So your skin cells, your epithelial cells that are within your intestine and whatnot, that there's a foreign invader present. Protect yourself. And so I may have you guys know some of the cytokines in tomorrow's lecture, but just for today, understand that that's what these molecules are for. They're meant to sound the alarm that, upregulate certain proteins so you can go ahead and not get infected. So that's going to be the main function of cytokines. The other main function that cytokines also have is to communicate with other immune cells on what to do. And how I mentioned with the macrophage slide, there's pro and anti-inflammatory cytokines, and these are going to have they're going to have the effects that, what their name suggests. If it's a pro-inflammatory response, then that's going to help create an unsuitable environment for whatever pathogen is invading. Versus an anti-inflammatory cytokines, those are going to basically tell the immune system, stop doing whatever you're doing. The pathogen is clear. Go ahead and facilitate tissue repair. Chemokines, on the other hand, they're like cytokines. Their structures are a little bit different. But chemokines are going to be molecules that recruit other immune cells to the site of infection. And the whole point of this is to, clear off whatever the invading pathogen is, strength in number. And so the more immune cells that you have in a particular location, the faster and more efficient you're going to be able to kill off the pathogen. But I'll end up talking about this tomorrow. Too many immune cells in a location can be detrimental to the patient. As highlighted with COVID, how I mentioned, it's not the virus that kills the patient, it's the immune response. It's because there's too many immune cells that are present that are recruited through these chemokines. And once the immune cells are present, then they secrete these cytokines that basically kill off cells that are present. And if you have too much of this tissue destruction, then that's why a lot of patients that end up getting COVID or severe cases of COVID end up on ventilators and subsequently dying, unfortunately. I like to describe chemokines as a perfume for immune cells. So basically, perfumes smell good, and we like to go to wherever where the perfume's most concentrated at. And so immune cells like to do the same. They like to go, like, there's going to be chemokines that are floating within the blood, but they're going to be very concentrated in the site of infection, or the site wherever the immune system is telling some of these other immune cells to go. And so that's basically the whole process or the whole point of chemokines presence, all for recruitment of immune cells. So understand these two. And so here I have a nice diagram of this whole process that happens when say a bacterial infection is going on. So here you have a bacteria that's phagocytosing bacteria. Because it's recognizing that there's pathogen present, there's also pathogen recognition receptors that are present on the surface of this. It's going to end up secreting chemokines and cytokines. And so what the chemokines are going to do again is recruit immune cells, like I mentioned before. There are tons of cytokines that are present, and in tomorrow's lecture, I'll probably highlight like a couple of really important ones that you should know. But in the context of this class, whenever I'm referring to cytokines, the majority of them that I'm going to refer to are pro-inflammatory. But they also have, another function, and one of the function is to basically, cause this thing called vasodilation. So it's going to cause your blood vessels to end up expanding, so you can get more blood into that particular area. So if you think of like when you have an infection on your skin or something and it starts to swell up red, well, it's because your innate immune cells, such as your macrophages, are producing these cytokines and chemokines to cause this vasodilation. And the whole point of this vasodilation is so you can help more immune cells to get to the site of infection. So typically you have your blood vessels are lined with these cell types called endothelial cells, and they form these nice tight junctions. So it's kind of difficult for cells to actually penetrate those tight junctions and get through to the other side. So what some cytokines are able to do is signal to some of the receptors that are on these endothelial cells and cause these tight junctions to separate a little bit. And that's what's known as vascular leakage. And so when you do that, it makes it easier for cells to migrate through these tight junctions. Once you have enough immune cells that are present, and this is through, again, chemokines and cytokines, then it's going to end up, trying to clear off the infection. And they're going to end up producing other inflammatory mediators that are going to end up causing pain. So that's why when it's swelled up red and you touch it, there's pain. It's because some of these innate immune cells are secreting these molecules that are causing that pain. They don't want you to, your body's telling you don't touch this area, leave it alone so the immune system can do its job. And then what is also another event that occurs, especially when you get a cut on your skin or something, are these blood clotting factors. And so the whole point of this is to seal off some of the circulatory system so the pathogen, the bacteria, can't circulate throughout the body. Because once any pathogen makes it to your bloodstream, like the main like circulatory system through your heart and lungs and stuff like that, a lot of the time it's game over unless you seek, therapy in the hospital where they can give you supportive therapy to, dampen the immune system, give you antibiotics or antivirals, whatever the infection is, to kill off the pathogen. But once a pathogen is in the blood, it is very detrimental to the patient. So your immune system is very good at basically walling that off so that doesn't occur. Yeah, how does ice help with inflammation? Basically, the coldness itself is, oh, so let me back up. So I mentioned that cytokines can cause this thing called vasodilation, or it causes your blood vessels to expand. Well, there's also heat that's being radiated from that. That's also helping with that expanding of the blood vessels. With when there's swelling or something, basically people will say, put ice on it to dampen it a bit. And that's going to end up basically closing some of these tight junctions a little more, because the coldness will actually cause it to contract a little bit. And, that's just more of a measure for people to, so there isn't like an over response to whatever the pathogen is. It's just to kind of keep it more mellow. Because there are some people that are more sensitive to certain infections, and if they're more sensitive, then that means your immune system is more sensitive, and if their immune system is more sensitive, then sometimes their immune system might go into overdrive. So that's how ice kind of helps with the swelling. It cools down the area, and then it and doesn't necessarily close these tight junctions, because some of these cytokines are keeping them open, but it makes the gaps between them smaller. I don't know if ice would be a bad idea. I think it really depends on how big the side of inflammation is, honestly. If it's not super big, then it can go either way. I think it's just going to be more to help alleviate you and the pain than anything. Your immune system's already activated. The pathogen's present. The immune system is going to recognize it and mount its response. So I think the ice isn't really doing anything more for the immune system. It's more of helping you, cope with the pain or whatever feeling you're experiencing. But I don't think it's gonna have a bad or negative effect. It's more for comfort than anything. So here I have an overview of basically bacterial clearance and how the adaptive immune system is implicated in this. So in the last slide, I kind of highlighted some of this that was going on here. So, we have a local site of infection. You end up getting a cut on your arm, and that introduces some bacteria to the upper surfaces of your epidermis. And you have some immune cells that are actually living in some of the layers of your skin already. So instead of having, cells circulating in your blood, you have these immune cells already at some of these barrier sites, so that if the barrier sites ever penetrated, then they can immediately respond. And so two of these main ones are the macrophages and dendritic cells. And so if they live in a particular tissue, then they're going to call tissue dendritic cells or skin dendritic cells. So wherever they're living. And so, like I mentioned before, macrophages, garbage collector of the immune system, they're going to try to uptake these pathogens to kind of basically prevent it from spreading. And then what you're going to have with the dendritic cells is they're going to take up some of this bacteria. Either they're going to phagocytose it as well and then fragment it within their own body, or they can, so you see that they have like these like finger-like projections. They can actually hug some of the pathogen, and I think that's called penocytosis. Basically, they can either, carry the pathogen that way by hugging it or by eating it and then cutting it up into pieces. And then they end up taking it to a secondary lymphoid organ. And so the most common secondary lymphoid organs people will typically refer to when it comes to immunology is the spleen and the lymph node. And so here we have an example of a lymph node. And I haven't decided, I don't think I'm going to have you guys know the different regions of the lymph node. Just know like this is going to be the site where your adaptive immune cells mainly live. You have adaptive immune cells that are circulating, and those tend to be naive or memory cells. But in your lymph nodes, it's mainly going to be B and T cells waiting for some innate immune cells, such as your dendritic cell or your macrophage, to come in and present antigen. Once the antigen is present, then the B and T cells can clonally expand. So again, that just means the cell can duplicate itself as many times as it needs. And then it'll end up leaving the lymph node or secondary lymphoid organ. So it depends on where it's at. Again, we have it in tonsils, in our gut, et cetera, et cetera. So once the T and B cells get activated, then they're able to migrate out of the lymph node and go into the blood, and then basically go to the site of infection. Again, chemokines are going to be the main players as to how these T and B cells are recruited to the site of infection. So again, the chemokines are going to be the perfumes, and then cytokines are going to open up some of these tight junctions in between the skin cells so they can go ahead and squeeze right through. And then once you have your B and T cells present, basically your B cells will secrete antibodies. So here we have an example of that Y-shaped antibody that are going to be able to bind to the bacteria. And then if any bacteria are intracellular, because you have two kinds, you have bacteria that infect cells intracellularly and some that just stay on the outside. So if these are actually inside the cell, then your T cells will come and basically kill those infected cells. So this is a basic overview of what happens in a typical bacterial infection and how it's cleared with both arms of the immune system. So tomorrow we're going to be focused on adaptive immunity. And again, just to highlight, this is where all the adaptive immunity and the education of the adaptive immune cells begins at the site of secondary lymphoid organs. So I'm going to go ahead and leave it off there. I feel like that was a lot of information. Hopefully you guys are able to digest most of it. I just try to layer it on as best as I could to, not confuse you guys. I hope, this is a good intro for you guys to understand immunology. Tomorrow, again, will be the main focus of how B and T cells are able to recognize pathogen and what they're able to do for the innate immune system. And then I'll highlight viral infections in particular, just so we have an understanding how our body is mounting a response against COVID. I also have some extra slides in there, just for more visuals for people, because sometimes I think the visuals I have on here are pretty decent, but sometimes people need multiple ways of seeing this representation of what's going on. So so feel free to have a look at those. It's all things that I've highlighted already in the lecture. I'll go ahead and release the lecture quiz in a bit. I went ahead and put in the sign-in sheet into the chat. So at this point, you guys are good to go. And if you have any questions, feel free to stay on and I'll clear up any confusion.